Okay,
Okay, please
great.
go ahead.
Okay, I'm Virgil from Institute and Plant and Microbial Biology, Academia Sinica, Taiwan. I would like to thank the organizer to give me this opportunity sharing my work entitled Arabidopsis AGB1 participated in the salinity stress response through bZIP17-mediated unfolded protein response. So the plants are constantly challenging by many environmental stresses, such as heat, cold, drought, also the pathogen attack. And all these affect the cellular homeostasis. And one of the very crucial organelle, which is endoplasmic reticulum, or called ER, was stressed and by this kind of the environmental change. And that is because the ER inside was accumulated overall many unfolded proteins. And this state and the protein was folding environment is tightly monitored. And also one of the key indicators is the status of the unlinked glycosylation of those nascent peptide. And like many other eukaryotic cells, plants has been utilized two of the UPR pathway to cope with the ER stress condition. So to cope, plants activated two of the pathway. Well, first one is IRE1, and this ER sensor was localizing on the ER membrane, which is equipped with either kinase as well as the endonuclease activity. So under ER stress means accumulated of unfolded protein. They undergo unconventional splicing of the bZIP60 transcription factor mRNA, and the spliced bZIP60 protein will enter to the nucleus and the activated unfolded protein response. In other words, is able to accelerate the protein folding capacity inside the ER. And this IRE1 this also can undergo read, which is a regulated IRE1 dependent decay, which will selectively decay the specific messenger RNA such as PR4, PRX34, which the purpose is to reduce the overall the protein translation needed in the ER to reduce the ER stress. And also the second pathway, which is achieved by the ER membrane titer transcription factor. They are bZIP17 and 28. Under stress condition, they will move from the ER to the Golgi apparatus, undergo regulated proteolytic processes performed by S1P and S2P. After that, the domain of the bZIP17 or 28 spliced by bZIP S1P and S2P will move into the nucleus and activate the UPR response. Different from any other mammalian system, plants equip their own way to cope with ER stress. There are some of the transcription factor localized on the plasma membrane, as well as the heterotrimeric G-beta protein was known to involve in the UPR regulation, which is quite different from the mammalian system. And talk about the heterotrimeric G protein. It has been known will respond to the environmental external stress by receiving the external signal via the G protein coupled receptor, in short GPCR, and it will initiate the interaction between G alpha, G beta, and G gamma. That by applying the chemicals called tunicamycin, are able to block the unlinked glycosylation on the nascent peptide, will able to block the protein folding inside the ER to induce ER stress. So in the assay, when we utilize the wild type and also other tDNA mutant of the G protein, we found that under most conditions, the seedling of AGB1 mutants was relatively larger than others. However, in presence of the tunicamycin containing plate and grow for 14 days, that they are severely sensitive to the ER stress, suggesting that AGB1 G-beta protein is essential for the response of the ER stress. And it was known that many other abiotic stress will trigger ER stress. One of the very typical abiotic stress is soil stress. And we all know that the soil salinity is one of the major threats for food security. For example, that every day, there are over 2,000 of the hectares of those cultivated lands in the US has been lost due to the soil salinity. And in other words, it actually caused more than 27 billion US dollar loss per year. And then in response, then under the soil stress condition, it will give the negative effect to the plants, such as reduced germination, inhibit the growth, and also reduce the yield. Even it will accelerate the leaf senescence due to the dehydration as well as the excessive amount of the sodium influx. So under this circumstances, one of vital hormone we know that is ABA will trigger the downstream signaling to initiate the upregulation of those transporter antioxidant. The purpose is to compromise, to mitigate those negative effects on the photosynthesis, protein synthesis, or water uptake generated by the soil stress, aim to restore the ion homeostasis as well as the osmotic adjustment to enhance the stress tolerance for the plant. And it has been previously intensely studied that AGB1 function in both soil stress as well as drought stress response. For example, in the G protein internectal, over 434 proteins, they are overrepresented. They are involved in the soil stress response. So suggesting that the overall G protein signaling is correlated to the soil stress. And also in the previous study suggests that in the microarray data using the AGB1 null mutant, they found that originally upregulated under soil stress, those antioxidant or transporter are significantly compromised. And in other data suggests that AGB1 itself will negatively regulate the expression of such as ABA1 or ProDH to increase the ABA or protein biosynthesis, or even negatively regulate the kinase MPK6 to involve the drought tolerance response. Furthermore, in the previously study suggests that the microarray data, AGB1 itself will contribute to the ion flux as well as transpiration. All these data suggest that AGB1 is involved in the soil stress as well as drought stress response, which is likely at the transcriptional level. When we talk about the transcriptional regulation, we have to talk about the bZIP transcription factor. And previously, several lines suggest that AGB1 interact with the bZIP transcription factor. This transcription family, which is composed by this basic region for DNA binding as well as the leucine zipper for the hetero or homozymer dimerization. And then there are several clades has been of the bZIP transcription factor being studied related to the unfolded protein response or the salt and osmotic stress response as well as light signaling. And this, like the previous data suggests that AGB1 is physically interact with the bZIP17-mediated binding and also the high-fi and high-edge interaction. Even for the high-fi itself has been known that it will compete the DNA binding with the bZIP28, one of the UPR sensor, suggesting that in the group of the bZIP family, they are able to crosstalk between two of the different transcription factors involving different kind of abiotic stress. And the interaction of AGB1 with those bZIP17 suggests that bZIP transcription factor is a good target to study AGB1 function involved in the transcriptional regulation of soil stress response. So to study this, we are previously able to generate one of complementation line transgenically express our genomic AGB1 sequence followed by the venous fluorescent tag in the AGB1 null mutant. And in the long-term 2 week of the soil stress tolerance assay, this transgenic line AGB1-V, the AGB1 venous are able to fully complement the hypersensitivity of AGB1 null mutant under the high salinity stress. And it will perform as good as wild type, suggesting that this is a complementation genome biologically and biochemically. So we take the advantage of these transgenic lines. Next, we want to study whether we can visualize AGB1 subcellular localization, for example, in the nucleus, to study whether they are able to contribute to the transcriptional regulation. For example, here, 7-day-old seedlings of AGB1 Venus treated with high salinity stress, you can observe that under mock condition, they are expressed the network-like structure close to the cell periphery. However, when you given the 1 hour, 2 hour, or 4 hours, you start to observe AGB1 Venus would show a punctate-like or duct-like structure in response to the salt stress. To further study whether this subcellular localization correlate to the nuclear localization, for example, we are constant with these transgenic plants with the plasma membrane marker, FM464, ER tracker, as well as DAPI. And all these suggest that the relocation of AGB1 Venus may locate it to the nucleus in response to the 4 hours salt stress treatment. We further isolate the biochemically of the nuclear fractions to compare here that the 4 hours intensity of AGB1 Venus is significantly increased compared to the 0 hours of the AGB1 Venus protein accumulation. And these data suggest that under stress response, AGB1 Venus relocated to the nucleus, probably for their function involved in the transcriptional regulation. So next, to study this transcriptional regulation, the potential involvement of AGB1, we performed the microarray analysis using 7-day-old of the wild type and the AGB1 mutants roots. And we are targeting for those 22 of the genes, which is known to as a downstream of bZIP17-mediated salt stress responsive gene. And you can see that in the heat map compared to the wild type. Overall, these 22 genes are downregulated in AGB1 mutants. To further validate this data, we also performed RT-qPCR. Then these 6 selected genes, such as NEC19, MIM75, RD20, LTP3, DR7, and TSPO, they all are downregulated in AGB1 mutants in response to the high salinity stress. As a control, you are able to observe that their salinity induction in AGB1 bZIP17 mutant was compromised. And interestingly, this is also inhibited in the double mutant of AGB1 and bZIP17. Taken together, suggests that AGB1 at least partially involved in the bZIP17-mediated salinity responsive gene upregulation. So to test one of the possibility for how they crosstalk each other among AGB1 and bZIP17, we first performed the genetic to generate double mutant, and then these to assay their interaction between AGB1 and bZIP17. First of all, in the germination assay compared to the black line, which is a wild type, in the both mock and also salt stress condition, wild type is germinated overall better compared to AGB1 bZIP17 single mutant as well as AGB1 bZIP17 double mutant. In the long-term stress condition assay for 2 weeks, and together with the quantitative analysis, we are able to observe the reduction of the bZIP17 under high salinity was further enhanced in the presence of the AGB1 as a double mutant, which phenocopied to the AGB1, suggesting that they have their certain crosstalk genetically between each other. So next, since we already showed that the nuclear localization of the AGB1 and also the genetic interaction between AGB1 and bZIP17, even more, the bZIP17 downstream gene expression in response to the salt stress was compromised in absence of AGB1. We wonder whether AGB1 physically interact with the bZIP17. So first of all, in absence of the bZIP17 does not affect AGB1 nuclear localization under salt stress, suggesting that relocation to the nucleus of the AGB1 does not depend on the bZIP17. And also under salt stress condition, the AGB1 itself are able to co-localize with the bZIP17 in the nuclear fraction. In the biopsy assay, we are testing the physical interaction in the Arabidopsis mesophil cell. that we are able to show that by the different reversible combination of the AGB1 and bZIP17, we show the physical interaction of them in planta, suggesting that they have the potential to interact physically and genetically under soil stress condition. So lastly, what I would like to address the very important question is that whether the reduction of the bZIP17-mediated gene induction in absence of AGB1 is due to the effect they are affected by the S1P, S2P mediated processing of the bZIP17. So we generate a transgenic line in the background of the bZIP17 null mutant that the genomic bZIP17 will N-terminally fuse with the mRNP red fluorescent protein driven by its own endogenous promoter. Compared to the zero condition, we are able to observe increased amount of the nuclear localization of bZIP17 messenger mRNP, and suggesting that this is a good reporter to read the bZIP17 readout of the nuclear localization under soil stress condition. We further take the agrobus, that is a change-in assay expression system, to express this construct in the wild type S1, S2, S1P, and S2P double mutants, and also AGB1 single mutant to observe the nuclear relocalization of bZIP17 in presence of the soil stress condition in the different genetic background. Compared to the Columbia wild type, S1P, S2P serve as a positive control. You are able to observe the similar nuclear localization in the both stress and the non-stress condition, suggesting that this relocation is regulated by S1P, S2P mediated proteolytic processing. Interestingly, in absence of AGB1, the nuclear relocalization of bZIP17 are compromised, suggesting that the potential contribution of AGB1 to mediated S1P, S2P processing of bZIP17 under soil stress condition. Lastly, this schematic model summarize our study recently under normal condition, under wild type in the soil stress condition, that AGB1 is shortly relocated into nucleus and probably contribute to the relocation of bZIP17 from the ER, go to the Golgi, undergo proteolytic processing, and release the nuclear located version of the transcription factor, which able to undergo transcriptional upregulation of those UPR genes, as well as salinity stress responsive gene, to help plants to cope with both ER stress and also soil stress condition. In comparison to AGB1 null mutant, we're able to observe the reduction of the bZIP17 relocation from the ER to the nucleus, probably as one of the contribution of the AGB1. They are not able to respond to the soil stress properly due to the downregulation of bZIP17 mediated gene expression. So in the future, we like to further address what's the potential contribution of those diverse substrate localization of AGB1 under various abiotic stress conditions. In the Taylor database, we noticed that there are splicing variant of AGB1, which generated by the intron retention as well as alternate splicing sites. So these are able to generate 4 splicing variant, and 3 of them are responding to the soil stress other than the last one. By generating the reporter line of AGB1 of those 4 splicing variant, we treat them with the soil stress. They are behaving differently in terms of the substrate localization, different from the 1.1 and 1.4. 1.2 and 1.3 have the diverse localization, including ER and nucleus. So we will, in the future, we will further address these contribution of the alternate splicing of AGB1 in response to the stress, including ER stress and the soil stress. Lastly, I would like to acknowledge Dr. Kazuya Kanehara and Dr. Yuki Nakamura for their advice, also my former colleagues for their suggestion and the contribution. Lastly, the financial support comes to the IPMB Academia Sinica, and I would like to take your suggestion, comment, and question. Thank you.